Okay, in this video, we'll be going over cholesterol metabolism. Sorry if I haven't been doing these as consistently. I have them all done, I just have to record them. And the last couple lectures, they're pretty long, so they take a while to make. Okay, so in this lecture, we will talk about cholesterol metabolism, how it's regulated, what it's used for, a lot of the stuff like that. So cholesterol metabolism starts with acetyl-CoA, which is similar to how ketone bodies, similar starting point as ketone bodies, similar starting point for like TCA, a lot of stuff. So we'll talk about how that's all affected. It happens in the cytosol. I don't think I mentioned that. Cytosol. And then we make a bunch of intermediates. You guys don't have to know that. All of these, just a bunch. Then we make squalene, which is important. And then we convert into other things and then eventually make cholesterol. I will mention it has multiple layers of regulation. So this is called de novo synthesis, which is how it's synthesized in the body. So that can be regulated. You can also take it up from your diet. So that's regulated as well. And then packaging, repackaging between different tissues can also be regulated. Okay, so like I said, we start with acetyl-CoA. Now we're starting to see a lot of pathways connecting. Acetyl-CoA is a start point for TCA. Remember acetyl-CoA, just briefly. Comes in with oxaloacetate, and then we make citrate. And then fatty acid synthesis starts with acetyl-CoA in the cytosol. Beta oxidation in the matrix ends with acetyl-CoA. And then cholesterol metabolism also starts with acetyl-CoA in the cytosol. Because both fatty acid synthesis and cholesterol start with acetyl-CoA and they both happen in the cytosol, they're usually right together. And we'll talk about a mechanism that the cell uses to upregulate both of them at the same time. Okay. Next, so this is what I was talking about with multiple layers of regulation. We can regulate the synthesis of it. We can regulate the uptake, packaging, and transport between the different tissues. And then the delivery to the organs after you repackage it and you transport it can also be regulated. So it's regulated on multiple levels. And it's really important because cholesterol plays a really big role in cell membranes. And it's important that we regulate the levels of cholesterol inside the cells. Okay, our cholesterol pool. So 1 gram a day comes from de novo synthesis. So this is synthesized inside the body. Inside the body. So this is what we'll talk about at the beginning using acetyl-CoA. And then 0.3 grams come from diet. And that gives us our cholesterol pool for the day, and then some of it gets excreted as well. Okay. So let's brief overview. So the first part, I mentioned it happens in the cytosol, but the first part occurs in the cytosol, which is acetyl-CoA to HMG-CoA. It's important that this happens in the cytosol because HMG-CoA is the same intermediate that we have in ketone bodies. You want to run cholesterol metabolism completely differently from when you want to run ketone bodies. You run ketone bodies synthesis when you have when you're in a low energy state. You run cholesterol synthesis when you're in a high energy state. So it's important to regulate between these two pathways because if you don't, then that could cause a futile cycle. So that's why cholesterol synthesis occurs in the cytosol and ketone body synthesis occurs in mitochondria. So this part occurs in the cytosol, and then after that, everything else occurs in the smooth ER. So this would be your cell. This is your nucleus. So the cytosol is right here, and then you have your rough ER and then like smooth ER. So after it's done in the cytosol right here, and then it goes into like on the membrane of the ER, and then it runs through all these reactions. So and a lot of these because it happens on the smooth ER, a lot of these reactions are a lot of these enzymes are membrane bound. We'll also see that a lot of the steps require NADPH. So that means when we run cholesterol synthesis, we need to be running the oxidative phase of the pentose phosphate pathway. And like I mentioned, HMG-CoA is the same intermediate as the one in ketone synthesis. Okay, so this is the first part, like I said. The second part happens in smooth ER. It's divided into two parts, and we'll talk about all of those. Okay, so the first part is going from acetyl-CoA to HMG-CoA. So this is that right here. So this is very similar to the first two steps in ketone body synthesis. So if you already learned that, you won't have to learn it again because it's the same thing. The first enzyme is called ACAT2, which takes two acetyl-CoAs, and then we make this, I think it's called acetoacetyl-CoA, something like that. And then we add another acetyl-CoA, and then HMG-CoA synthase takes that, and then we make HMG-CoA. First step is the reverse. It's the reverse of a thiolase step, which is the last step in beta oxidation. So this is the same thing we talked about with ketone metabolism. It's the reverse of the last step of beta oxidation. Okay, and this is the same as the one in ketone body synthesis. So if you already learned about that, you should be good to go. Should I go over it? No, I'm not. It will take too long. Okay. So then after that, after you make HMG-CoA, you have your main regulatory enzyme, which is called HMG-CoA reductase. Takes HMG-CoA, and then we make mavolinate. I think that's how you pronounce it. I'm really bad at pronouncing this stuff. This step requires 2 NADPH, so this is what I mean when we say we use a lot of NADPH. So we want to make sure the oxidative phase of the PPP is running. This is the first integral membrane protein in the smooth ER. So if you go back, it's right here. So this is the first step that starts the smooth ER. Okay. After we make that, we do a bunch of other steps. 2 successive phosphorylations. You guys don't have to know all the intermediates. So know up until, this is from my experience. I don't know what Dr. Kanopka wants you guys to know. So I would ask her. So for us, we had to know mavolinate, and then we had to know this one, isopentyl pyrophosphate. We didn't really have to know like the ones in the middle between that. So this is you do 2 successive phosphorylations, and we use ATP for those phosphorylations. So the first step uses ATP, and then it's done by a phosphoprotein transferase, and then second step also uses ATP. It's done by a kinase. And then we may use another ATP, and then this time we get a decarboxylation. And then this gives us the isopentyl pyrophosphate. And this actually exists as an isomer. And it's important we'll see that because we need both isomers for the next reaction. This also tells us that the cell needs to be in a really high energy state to synthesize cholesterol, which, like I mentioned, it's opposite of like ketone body synthesis, and it also happens whenever you synthesize fats. So whenever you're synthesizing fats and cholesterol, you need a lot of NADPH and a lot of ATP. So the cell needs to be in a high energy state for this to happen. Okay, so it's important that when we make the isopentyl pyrophosphate, we also make the second isomer of it, which is dimethyl allyl pyrophosphate. I'm pretty sure you have to know these two at least, in case you're asked about it. And then because the next step, diurnal pyrophosphate, requires both the isomers to make that. So we do a head-to-tail condensation, and then we take diurnal pyrophosphate, we make another head-to-tail condensation. We use 2 diurnal pyrophosphates for the next step, and then we make francial pyrophosphate, which is made by another head-to-tail condensation. And then we take 2 francial pyrophosphates. We do a last head-to-tail condensation, and then we get squalene, which is the last linear precursor in cholesterol metabolism. This last step, squalene synthase, uses NADPH. So this is what I mean by needing a lot of NADPH. So we used ATP in all of these. We used NADPH in HMG-CoA reductase, and then now we're using NADPH again in squalene synthase. Okay. So let's talk about the first condensation, just to briefly talk about the mechanism. So we have isopentyl pyrophosphate, we have dimethyl allyl pyrophosphate. We take both isomers. Pyrophosphate leaves from dimethyl allyl pyrophosphate, we make a carbocation. The double bond in isopentyl pyrophosphate attacks that, and then we get this intermediate. We lose a hydrogen, and then we make a double bond inside of the hydrogen, and then we get geranyl pyrophosphate. Okay, after we make that, we we take 2 geranyl pyrophosphates. We lose a similar mechanism to the one before it. Head-to-tail concentrations are all the same. Actually, they're not all the same, but they're all really similar. We lose a pyrophosphate, and then we get a franciol pyrophosphate. Franciol pyrophosphate is associated with protein prenylation. If you don't know what protein prenylation is, it plays a big role in the ubiquitation of protein. Ubiquitation just targets a protein for degradation. So this kind of links cholesterol metabolism and post-translational modifications. So whenever something, what I mean by protein prenylation, whenever it plays a role in ubiquitation, whenever something is tagged with ubiquitin, it's usually degraded. We make squalene, which is the last linear precursor. And then all of this still occurs is associated with the ER, like I mentioned. Okay. After we make squalene, we have an enzyme called squalene epoxidase. Before we go through that step, squalene cyclizes. We get 2 very close cyclohexanes. They're not cyclohexanes yet. And then we go through squalene epoxidase. We use so this is another step that uses NADPH. We use oxygen, and then we form this epoxide over here, which is very important because if you remember from Ockham epoxides, they're very high energy if you break them. So we were going to make this epoxide double bond. This oxygen that has bonds with carbons, so then we cleave in the next reaction, which helps us to cyclize the molecule even more. Squalene epoxidase is a cytochrome P450 enzyme. Usually these enzymes, whenever you see them, use NADPH. We reduce 1/2, so oxygen over here has 2 oxygen. It's a diatomic oxygen. It has 2 oxygen atoms. 1 oxygen is reduced in water. The other oxygen helps us form the epoxide, which is highly reactive. Okay, let's briefly talk about cytochrome P450 because I don't think we've ever talked about the mechanism, so it's important to know. We get our electrons from NADPH. NADPH gets oxidized to NADP. This is the reductase part of cytochrome P450. FAD accepts the electrons and then becomes reduced to FADH2. FADH2 then passes the electrons to FMN, which then becomes reduced to FMNH2. In that process, FAD is in the oxidized form. FMNH2 gives the electrons to the cytochrome P450 catalytic enzyme. 1 oxygen becomes reduced to water, and then the other oxygen is attached to a heme, and then it becomes this radical. Then the radical gives the last electron to our substrate, and then we form an OH bond, and then cytochrome P450's heme that has the oxygen, that has the stuff attached to it, returns back to its normal state. Okay, so after we make the 2,3-oxidosqualene, which we made in this step over here, we start protonate the ring, and then we start to do the ring closure. So the ring closure starts over here, and it goes through the entire molecule, so it starts from one end of the molecule to the other. Eventually, we'll lose the protons around the carbon number 9, and then we'll make a double bond. The mechanism of this is not as important. Then we get Lane-Stroll, which is an intermediate in the cholesterol metabolism. It's important because sometimes you'll be given cholesterol derivatives, or stuff that uses cholesterol, and a lot of them will look similar, but it's important to have this double bond at the carbon 9 position, because sometimes the molecule will look really similar. But if it doesn't have this double bond carbon 9, then it wasn't a cholesterol derivative. Okay, then we make a bunch of subsequent reactions from Lane-Stroll all the way down the cholesterol. We do demethylation, desaturation, saturation, and then eventually form cholesterol. A lot of them are unnecessary to know. You can look at the enzymes, look at the reactions, and have an idea, but they're all catalyzed by cytochrome P450, so it's a very similar mechanism to squalene epoxidase. Okay, so I'm going to briefly talk about the last reaction, which is 7-hydrocholesterol going into cholesterol. So the final step is 7-dehydrocholesterol going into cholesterol. I have nothing written in on it. Basically, 7-dehydrocholesterol can then be converted into vitamin D as well, which is different from cholesterol, which I'll talk about. But this is the final step. I think I mentioned it because I want to talk about the deficiency. If you have a deficiency in this reductase, you have this syndrome called semi-lamelli-Opitz syndrome. It's a developmental disorder. It causes autism. You can also have malformations in the heart, GI, kidney, and limb. I think that's the only reason I mentioned the last step. I think it's also important to know that the 7-hydroxycholesterol reductase takes 7-dehydrocholesterol and then converts into reductase. I think that one is one of the enzymes you have to know. Okay. All right, let's talk about the bioactive derivatives of cholesterol. So we already said francel pyrophosphate can be used in protein prenylation, which is involved in ubiquitin. It can also be converted into ubiquitin, so this is it can go through a bunch of intermediates and then eventually convert into ubiquitin. So this is electron carrier we talked about in ETC, another name for it. So ubiquitin would be the oxidized form of coenzyme Q. Coenzyme Q. So this is ubiquitin is another name for oxidized coenzyme Q. And ubiquinol is reduced coenzyme Q. We make the 7-dehydrocholesterol, which then can be converted into vitamin D. And then it's also, if it goes through dehydrocholesterol reductase, it can be converted to cholesterol. We can also make dolicol, I think that's how you pronounce it. It's from francel pyrophosphate, and it's an oligosaccharide chain of glycoproteins. It's used in that. Okay, so let's talk about vitamin D. So we said it comes from 7-dehydrocholesterol. So 7-dehydrocholesterol. So this reaction exists in the skin. If we have the sun, that's why they always talk about the sun giving vitamin D, because the sun catalyzes the reaction between 7-dehydrocholesterol to vitamin D3 in the skin. Vitamin D3 then goes to the liver. This reaction can also happen from dietary intake. So if you eat vitamin 2 can go into the liver and run in the same pathway as the vitamin D3 pathway. 27-hydroxy vitamin D is then formed in the liver. You guys don't have to know the reactions of how that forms. Just know that 25-hydroxy vitamin D is formed in the liver. It's shipped to the kidney, and then the kidney makes 1,25-dihydroxyvitamin D2, which is another name for vitamin D. So that's kind of how vitamin D forms. It can form in the skin, or you can get it from vitamin D2 in a diet. You make 25-hydroxyvitamin D in the liver. It goes in the kidney, and then the kidney makes 1,25-dihydroxyvitamin D2, which is another name for vitamin D. Okay, vitamin D can then bind. So vitamin D has a lot of effects, but a big thing that it does is it changes transcription in the tissues that it works in. So it can bind on the VDR receptors. They dimerize and then translocate into the nucleus. They form a VDR RXR complex. So this is the 125-dihydroxyvitamin D2, which is vitamin D. It binds its receptors, dimerizes, and then we get this complex called VDR, which is the vitamin D receptor, RXR complex. It binds this vitamin D regulatory region called VDRE, and then this can change transcription in the genes. The genes that it changes transcription in is tissue dependent. So, for example, in vascular wall, it it induces vascular smooth muscle cells differentiation, or vascular endothelial growth factor synthesis. And then in cardiac cells, it can regulate the cell cycle, and then it can help with synthesize ANP. If you don't know what ANP does, it's released when you have, when you have high blood pressure, and it helps lower that. It can also help with fibroblasts and MMB balance. Fibroblasts are cells that release collagen. Just extra information. So let's talk more about the bioactive derivatives of steroid hormones. So we already another bioactive derivatives of cholesterol are steroid hormones. So we talked about vitamin D. We talked about prenylation. I think talked about dolicol or ubiquinone. So another one is steroid hormones. That's where you get your cortisol, aldosterone, testosterone, and progesterone. Cortisol is released from the adrenal cortex. It's your major stress hormone. Aldosterone is also released from, I think it is, I'm pretty sure it's released from the adrenal cortex as well. And it's released when you have low osmolarity in your blood, so it helps increase the osmolarity in your blood. Testosterone, of course, induces the male primary and secondary characteristics. Actually, it's just secondary, I think. And then progesterone helps in the menstrual cycle in the female reproductive system, of course. So this is cholesterol. I don't think you have to know all these reactions, but eventually you can make aldosterone through all these reactions. You can make cortisol over here, and then you can make testosterone as well. And you can also make dihydroxytestosterone. There's a bunch of other ones too, but I mainly highlighted these because these ones are the ones I know and the ones we go over as undergrads. Okay, uptake of cholesterol. So we already, if we go back, I'm not going to go back, it's a bunch of slides. So we already said that cholesterol can be synthesized. You get 1 gram a day when it's synthesized, and then 0.03 grams a day come from diet. So now we'll talk more about diet. So cholesterol is uptaken into intestinal cells first. A transporter called NPC1L1 has a sterol sensor, and then it uptakes cholesterol, binds to that. So this is the protein over here. Cholesterol binds to it, and then this induces endocytosis. And then you repackage cholesterol into a chylomicron, which can then go into the lymphatic system, and then can go into circulation. Before we send them into the lymphatic system, we can't just send them as cholesterols. They need to be converted to cholesterol esters. This is done by an enzyme called acyl-CoA cholesterol transferase, ACAT. So this is a group of enzymes. I have one specific that we'll talk about. Basically, they take cholesterol and then make cholesterol esters, and then they're put into chylomicrons, and then those can go into target tissues or the liver. They're mainly going to be sent to the liver because that's going to be your major regulatory organ. The cholesterol esters can bind to microsomal triglyceride transfer protein, which puts sterols in the ER membrane. So this helps put stuff in the ER membrane. So this is ALCAT. It's an example of ACAT. So ACAT is the ones that make cholesterol esters. Basically what it does, so this is lecithin, or another name for it is PC. Takes cholesterol, takes PC, makes cholesterol ester. We get the ester from the PC. So PC functions as a carbon donor in this case. Yeah, and then this acyl donor it can also function as an acyl. It's not a carbon donor, it's an acyl-CoA donor. It can also function as a acyl-CoA donor for chylomicrons. So to make chylomicrons, you need a lot of tags and then VLDLs, which we'll talk about in a little bit. So chylomicrons transfer cholesterol in tags, but they're also made of tags. So this is what I mean by KSO-A donor for chylomicrons. So to make the chylomicron, you need make a bunch of tags. So you can get the acyl-CoA donors from PC as well for chylomicrons. And then to make the species in VLDLs, you can also use PC as your acyl-CoA donor. Okay, chylomicrons. So remember we talked about in fatty acid uptake, they help repackage tags and cholesterol esters into chylomicrons, and then they ship them to the liver. So this is another way that fatty acid metabolism is linked to cholesterol. So remember, if you have something that inhibits chylomicrons or formation or something like that, this will affect both fatty acid synthesis and chylomicrons. Lipases then remove the tags from chylomicrons, and then you have cholesterol esters sitting in there, which are then shipped to the liver. This is a chylomicron. This is VLDLs, which I'll talk about what VLDLs is in a sec because I have a slide on it. But they both help transfer different cholesterol, different lipids throughout the body. Okay. I have it right here. So they're all called apolipoproteins. They're made of proteins, and then they're also made of lipids. So that's why they're called apolipoproteins. So chylomicrons, they're made of tags. They exist in the small intestine. They distribute tags and cholesterol to target tissues or the liver. VLDLs, they originate in the liver, and they distribute tags from the liver to target tissues. And those ones are also made of tags. So this is what I mean by chylomicrons and VLDLs get their acyl-CoA donors. Because they're made of tags, they get their acyl-CoA donors from PC as well. LDLs, low-density lipoprotein. This is very low-density lipoprotein. This is high-density lipoprotein. Low-density lipoproteins also originate in the liver. Those ones distribute cholesterol from the liver to target tissues. So VLDLs distribute tags to target tissues. LDLs distribute cholesterol to target tissues. Those ones are made of cholesterol. HDLs, so those ones also originate in the liver. So they originate in the liver, but they help return cholesterol in the liver. And I'll talk about it briefly of how that happens. So those ones return cholesterol to liver. They're made of cholesterols and PL. And they have different names depending on the percentage of protein. So because these are high density lipoproteins, so they have more lipoproteins, they have a higher density of lipoproteins, so that's why they're called high density lipoproteins. Those ones have lower density of lipoproteins, so they're called very low density lipoproteins. So this is density of lipoproteins. Okay. So this is your liver. This is target. So we said LDLs help distribute cholesterol from the liver to target tissues. So once the liver ships them, they're in LDLs. They bind on something called LDL receptor. This helps cholesterol uptake into the cells. So cells that want cholesterol express the LDL receptor, and then this is an example of receptor-mediated uptake of cholesterol. Once LDL binds into its receptor, this causes endocytosis, and then you harvest your cholesterol inside the cells, and then the LDL receptors, lipodensity lipoprotein receptors, are then recycled to the surface of the cell so they can grab more LDL particles inside the cells. Okay, so let's talk about LDLs, HDLs, all that stuff. I also want to talk about ABCA1 transporters. Okay, so this is intestines over here. It's not shown in diagrams. It sends the chylomicrons. Chylomicrons go through a bunch of stuff, but they're eventually sent to the liver. So let's just go from here. Then the liver takes these cholesterols, repackages them, and then sends them to whatever organs need the cholesterol. We actually originally send them into VLDLs. They run through something called lipoprotein lipase. So those lipoprotein lipase, they turn the VLDLs into LDLs. Once they're in LDL, once the cholesterol is in LDLs, they bind to LDL receptors at target tissues. Target tissues, and then you have endocytosis. You take the cholesterol inside the cell. You can use it in whatever you want. We use it in membranes. Use it to make steroid hormones. Whatever the cell wants. The LDL receptors then go back on cell membrane. ABCA1 transporters take cholesterol out of the target tissue and recycle them out. So this is what I mean. Actually, let me finish the cycle, then we'll talk about it. And then you have the ALCA, which take cholesterol and turn them into cholesterol esters. We put them in HDLs, which originate in the liver. So HDLs are right here. They're sent out of the liver, and they wait in the blood to accept cholesterol esters from target tissues. Once they accept cholesterol esters, they bring them back into the liver. And then the liver can do whatever it wants with them. So this is what I mean by another method of regulation. So we haven't talked about regulation yet, but how much LDLs that are expressed and how much HDLs the body has can help regulate how much cholesterol is going to other cells. So for example, if we have a lot of LDLs, there will be a lot of cholesterol in target tissues. That's why high LDL is usually bad. If we have HDLs, they help take cholesterol and then send it back into the liver. So they help with that. And then high HDL is usually not as bad as high LDLs, because LDLs just give it to target tissues. If cholesterol accumulates in target tissues, it actually can cause cell death, and it can be really toxic. ABCA transporters, so how fast these ones transfer cholesterol out of cells, that can help regulate the cholesterol levels inside the cell. So these are all different ways, the levels of all this stuff. How much LDL receptors you have, how much LDLs you have, how much HDLs you have, how much ABCA1 transporters you have can help regulate how much cholesterol each target tissue is receiving. Okay, yeah, I was talking about that. Regulation of cholesterol. So this is what I mean by LDL uptake and HDL release. So LDL uptake and HDL release is another way to help regulate cholesterol. The one we'll talk about a lot is the regulation of HMG-CoA reductase. So this is the rate limiting step. So that can be regulated in different levels. And then regulation of intracellular free cholesterols through acyl-CoA cholesterol acyltransferase. So this is the ALCAT. So basically how much if you can't ship cholesterols unless they're in cholesterol esters. So if they're not in cholesterol esters, you won't be able to send them out. So the levels of ALCAT, the enzyme that makes the cholesterol esters, can help regulate cholesterol levels in the blood, in the cell, target tissues, whatever. And then this is what I was just talking about, LDL uptake and HDL release. If you have a lot of LDLs being sent out, target tissues will have a lot of more cholesterol. If you have a lot of HDL released from the liver, then that will help. If you have a lot of HDLs released from the liver, that will help reduce the amount of cholesterol in target tissues. So those are different ways you can regulate. So HMG-CoA reductase, so this is one of the ways it can be regulated through different things. The reason you have a lot of, like I mentioned previously, the reason you have a lot of different regulations on cholesterol is because cholesterol is really important. It's in membranes. It helps make you all the vitamin D, steroid hormones, all that stuff. So it's very important to regulate levels of cholesterol. HMG-CoA reductase can be regulated through different ways. So you can have feedback inhibition, which we'll talk about, control of gene expression, rate of degradation, and then post-translational modification, such as phosphorylation or dephosphorylation. Okay, so the first one we'll talk about is post-translational modifications. HMG-CoA reductase, when it's dephosphorylated, it's in the active state. And when it's phosphorylated, so this is phosphorylated, this is inactive. Okay. AMPK, so remember AMPK is activated by AMP. It's a master regulator. AMPK adds a phosphate on HMG-CoA reductase and then phosphorylates it, inactivates it. This is important because remember AMPK signals low energy. So when there's a lot of AMP, you don't want to run cholesterol synthesis, because cholesterol synthesis is one thing. The way to think about it is very similar to fatty acid synthesis. You don't want to make cholesterol or fatty acids when you have low energy in the cell. You want to focus on other things. So when you have low energy, you don't want HMG-CoA reductase to be running. So AMPK then inhibits it. You also have HMG-CoA reductase phosphatase, so this activates HMG-CoA reductase. So it kind of does the opposite of AMPK. So HMG-CoA reductase phosphatase is acted upon by an enzyme called phosphoprotein phosphatase inhibitor. So what this enzyme does is it inhibits the activator. Inhibits. Do I have a green? No, I don't have a green. Oh, actually, it could get green. So HMG-CoA reductase phosphatase activates, and then this PPI, the phosphoprotein phosphatase, it inhibits the activator. So this one indirectly puts HMG-CoA reductase in the inactive state. Phosphoprotein phosphatase, we'll see it a bunch in other enzymes too. I think we already saw it with glycogen. This enzyme is activated by cyclic AMP and pKa. So we haven't talked about it, but glucagon eventually leads to more cyclic AMP. So we'll talk about this when I talk about signaling and energy regulation. And then glucagon is released when you're in low energy state. So when glucagon is released in low energy state, you increase cyclic AMP. Cyclic AMP activates PPI, which inhibits the activator. So eventually, when you have glucagon, you increase cyclic AMP, and then PPI puts HMG-CoA reductase in the inactive state. This is important because when you have glucagon, low energy state, you don't want to be running cholesterol synthesis. Hope that makes sense, but when we talk about energy regulation, it will make a little bit more sense. Okay, so that's one. So post-translational modification is one way that it can be regulated. Insulin and glucagon can also directly activate and inhibit HMG-CoA reductase. So we already talked about how glucagon inhibits. Insulin has mechanisms of activating HMG-CoA reductase, which when we'll talk about insulin, we'll talk about that. So if we go back, you can regulate HMG-CoA reductase, or you can regulate LCAT, the enzyme that makes cholesterol esters. So if we go back, cholesterol being converted to cholesterol esters, this will help cholesterols being shipped to other tissues or other organs or whatever. So the levels of LCAT, cholesterol increase acts as a positive feedback on ACAT is a type of LCAT, just so you don't confuse. Cholesterol can activate that to make more cholesterol esters and then can be sent to other organs. LDLs take cholesterol. So if you have a lot of LDL receptors, they can take extracellular cholesterol and then put it inside the cell, so it'll be intracellular cholesterol. So if you have a lot of LDLs, you'll have a lot of receptor-mediated endocytosis, and then you'll be receiving more cholesterol inside the cell. And then cholesterol can then also feedback inhibit HMG-CoA reductase, which we'll talk about in a sec. So cholesterol can actually go back and inhibit HMG-CoA reductase. So if you have a lot of cholesterol accumulating inside the cell, you don't want to be running more cholesterol synthesis, so then cholesterol feedback inhibit HMG-CoA reductase. And I think I have it in the next slide. Okay. So this would be your example of feedback. inhibition. So this is HMG-CoA reductase. It has a catalytic domain, and it also has a regulatory domain. Your regulatory domain has a sterol sensing domain. Basically what it just means is it has a mechanism of sensing how much cholesterol is inside the cell. And the way it works is, so this is HMG-CoA reductase. You have the catalytic domain and the sterol domain. High levels of cholesterol then eventually bind to the sterol domain and activate it. Once the sterol domain binds cholesterol, it activates ubiquitin, and then ubiquitin comes in, tags HMG-CoA reductase, and as soon as ubiquitin binds to anything, it's going to be degraded. So ubiquitin binds to HMG-CoA reductase, and then ubiquitin targets it for degradation. So this is done by a proteome. So, okay. So if you have high cholesterol, you don't want to be running. If you have a high cholesterol, you don't want to be running HMG-CoA reductase. So it degrades the enzyme when you have high cholesterol. Okay. Another way to regulate it is by transcription. So the genes involved in fatty acid synthesis, fatty acid metabolism, cholesterol, and tags, these things usually want to run together or regulate it together. At least fatty acid synthesis and tag synthesis. I think these are meant to say tag synthesis, not metabolism. I mean, you can say metabolism, but synthesis is a more accurate word. So when you want to synthesize all these things, they usually run together. There's about 30 genes involved in all of their things being synthesized together. So the cell wants an easy way to regulate them together and upregulate them together, downregulate them together. So if you have high energy, you run all of them together. If you have low energy, you don't want to run all of them together. So all the genes have a sterol regulatory element, which is called SRE. You have a protein called sterol regulatory element binding protein, SREBP. It binds these. The one that binds the cholesterol and fatty acid metabolism is called SREBP12. So that one is the one that regulates the fatty acid metabolism and cholesterol. All right, let's talk about how it works. So this is SREBP. It has a regulatory domain. It's associated with different protein called SCAP and INSIG. So INSIG is not mentioned. SREBB is in the membrane, it's embedded there. So this is the ER membrane. Remember, if we want to regulate these genes, we need to be in the nucleus. So what happens is, so when cholesterol levels are decreased, this causes a dissociation from SREBP, from INSAG, and SCAP. Okay, so then SCAP, when everything is dissociated, takes SREBP and brings it to the Golgi. Once it's in the Golgi, you have a serine protease. This serine protease does two cleavages. So then SREBP dissociates from SCAP, and then it goes into the nucleus because it's a transcription factor. Goes into the nucleus, and then SREBP binds SRE, and then increases cholesterol metabolism and fatty acid synthesis metabolism. So I'll say that again. So SREBP is in the ER membrane with SCAP and INSIG. When cholesterol levels are decreased, SREBP and SCAP dissociate from INSIG. They go to the Golgi, and then in the Golgi, there's a serine protease that cleaves SREBP from SCAP, and then SREBP goes into the nucleus, binds the regulatory region SREE, just one E, not EE, and then increases transcription for genes for cholesterol metabolism and fatty acids. Okay, so like I said, why is cholesterol levels important to regulate? Why do we need like 7 different ways to regulate it? Because that's crucial for membranes. When you have at high temperatures, the membrane fluidity increases. There's more temperature, there's more movement, so we need a way to regulate that. So cholesterol helps decrease membrane fluidity at high temperatures. And then when temperature decreases, the membrane fluidity starts to decrease a lot, and then cholesterol helps increase membrane fluidity in that state. So because it interacts with pretty much any change in the membranes, it helps regulate the membrane fluidity. Excess cholesterol, so too little cholesterol won't help regulate the membrane fluidity, and then extra cholesterol can actually disrupt membrane proteins, and then this can cause cell death. So it's important to regulate the levels of cholesterol. Make sure there's just enough for the cell. Okay, so there's so this is very similar, not the same, very similar to ABCA1 transporters that excrete cholesterol. So it helps excrete cholesterols from cells that have a lot of cholesterol in the membranes. Because this cholesterol in membranes can be toxic, so it helps repackage them for transport. So repackage them for transport so they can be excreted. And then usually this is expressed in organs that do a lot of cholesterol metabolism. So we'll see this a lot in the liver, the intestine, macrophage, foam cells, because they have a lot of cholesterol in their membranes, so they need a way for them to regulate that so it doesn't cause cell death. So this is the lumen intestinal cells. You send cholesterol in chylomicrons, and then chylomicrons travel to the liver. In the liver can then repackage them into cholesterol esters. So CE is cholesterol esters. Cholesterol esters can then go into VLDLs. It can go into LDLs, and then our target tissue here is a macrophage. So LDLs go into a macrophage to help take up the cholesterol esters. Macrophage does whatever it wants with those, and then it sends the cholesterol esters back. So this is the mechanism of what I'm talking about. So ABCA1 helps send them back. So helps with the repackaging. So to send it back, if you have a lot of cholesterol inside the target tissue, you need those two proteins. So you need SOAT to repackage it. You need ABCA1 to send it. HDLs are released from the liver. HDLs come in, grab cholesterol esters, and then bring them into the liver to help decrease the cholesterol levels in the blood. So this is just a way to regulate cholesterol levels between all the different organs and the blood. So that's what SOAT is used for. Okay. So another example would be the brain, because 95% of cholesterol occurs in the brain is de novo synthesized. The reason the brain needs a lot of cholesterol is it uses it for membrane. So you have like your myelin sheath. Myelin sheath uses a lot of membrane, so it uses a lot of cholesterol. Some of the cholesterol in the brain is used in glial cells, and some of it is used in neurons. Basically what happens, let's just follow an example here. So we have cholesterol. This is the astrocyte. You have cholesterol being made. It's shipped out of ABCA1. It goes into the neural cell, and then the neural cell makes the form 24SOHC, which is 24-hydroxycholesterol, and then this is the form that it excretes it in. So other tissues can excrete cholesterol as cholesterol esters. The brain excretes it as 24-hydroxycholesterol. It's important because because the brain uses a lot of cholesterol for its membranes. It's important to have a mechanism for it to excrete. Then 24-hydroxycholesterol is the major form excreted. It goes into the liver. The liver uses that for bile acids. So this is actually how bile acids are mainly made. They're mainly made from the 24-hydroxycholesterol that's excreted from the brain. So this is another way to help maintain cholesterol homeostasis between all the different organs. If we don't excrete this, we'll have low levels of bile acids, which can then affect other things in the digestive systems. The liver uses this for bile acids, and then takes bile and then sends it into the gallbladder, where it's stored for later use. Okay. So this is why cholesterol is usually used for bile acid synthesis, because its major form, 24-hydroxycholesterol that's excreted from the brain, is what it's used for. So bile acids, just to summarize, they're essential for absorption of fats from diet. They help emulsify fats. What emulsify fats just means it allows lipoprotein comes in and hydrolyze them. That's what emulsifying fats just means. They're synthesized in the liver, stored in the gallbladder, and then they're also an energy source for gut microbiomes. The liver can actually send biles to gut microbiomes. They deconjugate them and then send them back to the liver. So I'll talk about that briefly. So bile acid derivatives. So this is another derivative. So we already talked about steroid hormones. We talked about vitamin D, ubiquinone. Franciol pyrophosphate can be used in protein prenylation. Another thing is bile acids. Those are derivatives of cholesterol as well. So they're produced by the host to uptake from diet, and then they're also modified by the microbiome. So it doesn't show it here, but this is the microbiome. These are the two. Those are the two bile acids in this picture, by the way. Deoxycholic acid and lithocholic acid. Okay, so basically the liver can send those bile acids to the microbiome. They can use it as energy source. They modify it and then send it back to the liver to use. And we'll talk about why it's important in a second. Why it's important for the gut microbiome to modify our bile acids. In this picture, it shows as well short-chain fatty acids, which we talked about briefly in fatty acid metabolism. They help link microbiome in the human organs. And then this is TMA, which is another thing that helps link everything, and it comes from carnitine as well. But we're just focusing on bile acids for now. Okay, so bile acid synthesis, let's talk about that briefly. They can be synthesized with two different pathways. But it's mainly uses cytochrome P450 enzymes or sterol 27 hydroxylase. Basically, what bile acids are used for is they're kind of sink for to get rid of cholesterol. So when we have a lot of cholesterol, we don't want to excrete it. So it can instead be used to make bile acids to get rid of the cholesterol. The two different pathways to make them. So they use both these enzymes. You can make them in the ER. This pathway, the neutral pathway, it's called the CYP7A1, so this neutral pathway. Or you can make them in the mitochondria. This is the acidic pathway. This is the CYP27A1. So you can make bile acids differently. In this picture, this uses the P27A1. This is the CYP27A1. And then this is how they start. So they can take cholesterol, and then we can go into the CYP27A1 or the CYP7A1. And then you eventually get deoxycholic acid or lithocholic acid. Okay. Eventually, they're conjugated in the liver, because the liver is what synthesizes the bile acids, to taurine or glycine. So this is taurine, and then this is glycine. Well, it's not really glycine because it doesn't have everything that glycine has, but it's conjugated to it. They're used as, again, a sink for cholesterol elimination. When you have a lot of cholesterol and you're going to get rid of some of it, you convert it into bile acids. And then microbiome can then further modify the bile acids to re-enter circulation. So when they're actually conjugated to cholate and taurocholate, you actually they're not usually the main form used by the body. So we need to send them to the microbiome where they can modify them so they can be used by the body. So what happens is we need to deconjugate them. We need to hydrolyze the taurine and the glycine conjugates that are made. So we have deoxycholic acid, lithocholic acid. They become conjugated to taurine, and then glycine. But then that's not the main use. That's not mainly how they're used. So they're sent to the microbiome, where the microbiome does deconjugation. And the gut microbiome, they hydrolyze taurine and glycine conjugates, and then eventually send them back to the liver. So this is the liver. This is the microbiome that exists in intestines. Liver makes bile, sends it to the intestine. Microbiome then modify them. They deconjugate them, and then they send them back through the portal vein from the intestines to the liver, and then the liver can then use these. And then we also said the bile acids can be used as an energy source for microbiomes as well. Bile acids can also activate adenylate cyclase and make cyclic AMP, and this can affect metabolism. Just add it in there in case you have a question on it. Usually 95% of biles that are sent to the microbiome are reabsorbed back into the liver. And then the ones that are still conjugated, those ones are excreted into the fetus. So the ones that are deconjugated sent back to the liver, and they can be used again. Or not used again, they can be actually start being used. And then the ones that are still conjugated, they're excreted, because we can't use conjugated bile acids that are made in the liver. We have to send them to the microbiome to deconjugate them to be able to use them. Okay, so this is called atherosclerosis. This is caused by accumulation of LDLs. Well, it's caused by smoking, high blood pressure, or cholesterol. Basically, what happens is you have accumulation of LDLs. This causes inflammation, recruits macrophages, and then the macrophages differentiate into foam cells. Foam cells are macrophages with a lot of lipid, and then the rupture can cause stroke and heart disease. So this is a picture of endothelial cells. You have the fibroblasts, mast cells. LDLs come in. The LDL come in, and then eventually you have this rupture from LDL accumulation, and then foam cells come in. These are I think these are macrophages in the picture. And then, but eventually, when you have a lot of macrophages that cause inflammation, then they become the macrophages, which are these ones, differentiate into foam cells, and then the foam cells have a lot of lipid, and then they can rupture and cause a lot of bad things. Okay. Statins, I'll talk about those briefly. They can be used for people who have high cholesterol, for patients with high cholesterol. Basically, what they do is they inhibit HMG-CoA reductase. They act as competitive inhibitors. So instead of HMG-CoA binding to HMG-CoA reductase, statins, they look very similar to HMG-CoA. They compete for that spot. They bind to HMG-CoA reductase, and they inhibit de novo synthesis. This is good for anyone with hyperlipidemia. So these are the different statins that can be used. So statins is just a good medication for high cholesterol patients. Okay. Briefly, we'll talk about bacterial metabolism. So most prokaryotics actually can't synthesize cholesterol, and they use what is called hopanoids. Basically, they fulfill the same function, but they're not considered cholesterol. You see they don't have the double bond at carbon 9. They also are modified a little differently over here. Hopanoids, that's what they're called. And intracellular pathogens can use host-derived cholesterol if the pathogens can make their own cholesterol. Okay. To summarize, to make cholesterol, we used acetyl-CoA, we used NADPH, we also used ADP, we make squalene, and then Lanstrol, and then we eventually made cholesterol. It can be regulated different ways. So we said we can regulate HMG-CoA reductase differently. Packaging and circulations, so that's VLDLs, HDLs, the levels of those LDL receptors, all the levels of that stuff can help regulate the cholesterol levels inside the cell, and then levels of free intracellular cholesterol can also help regulate the cholesterol because they can feedback inhibit. And I also didn't mention ALCAT. ALCAT can help make cholesterol esters. Apolipoproteins, very low density lipoproteins, low density lipoproteins, high density lipoproteins, chylomicrons. Know the different like functions for those, which ones bring stuff in. So chylomicrons bring tags and cholesterol to from the intestines to target tissues. LDLs help distribute cholesterol to target tissues. VLDLs help distribute tags to target tissue, and then HDLs help take cholesterol from target tissues back to the liver. And then you excrete cholesterol as 24-OHC, which is used to make bile acids. And then statins are used as competitive inhibitors to help inhibit HMG convertase. So that's it for this video. I'll try to upload it as soon as I can, and then I'll also upload the summaries for it. Thanks for watching.